Okay. So, we have just discussed the different techniques that are used to separate the proteins, and before that we have also discussed different techniques that are used to identify the structure of a protein. Now, let us do one problem to sort out any doubt or clarification or to reinforce our concept. Okay. Now, there is a problem that you are trying to determine the sequence of a protein that you know is pure. Given the most likely explanation, give the most likely explanation for each of the following experimental observations. Now, what are the observations? The Sanger's reagent, that means the 2,4-dinitrofluorobenzene or FDNB, that identifies two N-terminal residues, alanine and leucine, in roughly equal amounts. So, what does it indicate? It indicates that it has the protein at least have two chains, where there is one chain it has got alanine as the N terminus, and the other chain there is leucine, that is the N terminus of the other chain. This is your X chain and this is Y chain. Okay. And we will come back that how these are tied together, that is they are in the problem itself. Actually, the next is your protein has an apparent molecular weight of 80,000 as determined by SDS polyacrylamide gel electrophoresis. After treatment of the protein with performic acid, that means a formic acid which is further oxidized, that is performic acid HCO3H, that is what is performic acid. The same technique reveals two proteins of molecular weight 35,000 and 45,000. Okay. So, what is said here? First, if you do SDS polyacrylamide gel, remember what SDS does? SDS is basically denature the protein, and then after denaturation you get the, you do the gel electrophoresis, and then you also run a molecular weight ladder, and from the molecular weight ladder you can say that what is the molecular weight of your protein. So, SDS page says that the molecular weight is 80,000. Okay. On the other hand, if you treat the protein with performic acid, now I think that point we have not said anything yet, that is that if there are two chains of proteins and if they are connected by disulfide linkages, that means there is a cysteine here, there is a cysteine there. So, that is the way of connecting two chains together, to separate chains together, that is usually through disulfide bonds. Now, the question is, if you treat this one with performic acid, that is HCO3H, then what happens? This disulfide bond breaks and this becomes a sulfonic acid, and this the other chain also becomes a sulfonic acid. That means, the chains are now separated, that is the most important thing. So, what happens when you did the SDS, this SDS page, at that time you get only one band 80,000, okay? That is the denatured protein. But when you did with performic acid, then you see that this is actually made up of two chains, one is 45,000 and the other is 35,000. These are the molecular weights. That means, your X chain, suppose the X is 45, then Y is 35 or vice versa. So, now you have a rough estimate that after performic acid treatment you get these two bands. That means, these 45 and the 35 are connected by disulfide, at least one disulfide. There may be more than one disulfide also, it does not say how many disulfides are there, but at least the minimum structure that you can write here is that these are the two structures, two chains, and this is alanine and this is leucine, and suppose this is the 45,000 and this is the 35,000. But I again repeat, this could be the other way around, this could be 35 and this could be 45, because the information is not given that what is the N terminus sequence of the 35 or what is the N terminus sequence of the 45,000, that is not shown. Okay. So, we have to write this or the other, these other ones. The last one is size exclusion chromatography, that is called gel filtration, we have learned that. Size exclusion chromatography indicate that the native protein has an apparent molecular weight of 160,000. Now, size exclusion chromatography does not disrupt any structure of the protein. Whatever protein structure is there in the native state, that is retained in size exclusion chromatography. So, that means if the protein is multimeric in nature, then that multimer will remain as one unit, and when you do size exclusion chromatography, depending on their rate of elution and the cut, that means the value where you I told you that the gel has a cut point, that this up to this molecular weight it will allow to be retained, or this will allow to be passed through. So, from that the most important is that means the native structure of the protein, the molecular weight is 160,000. So, how do you get 160,000? That means it immediately tells you that the protein must be composed of two sets of this alanine leucine structure. So, the actual protein must be alanine and then another chain, this chain, and then leucine, then this chain, they are connected by this disulfide, and then these two proteins are having some interaction to make a dimeric nature of this. Okay. So, basically this is a tetramer, but dimer of a heterodimer, because this is a heterodimer, and then this heterodimer is again dimerized. So, actual protein is dimer of a heterodimer, that means that is a tetramer. So, the actual protein is tetramer. So, you see this, that development of different techniques that will ultimately see. Suppose if you only do size exclusion chromatography, you will not be able to know that there are this the tetrameric structure, that you will not be able to tell at that time. So, you have to do SDS page, you have to do performic acid, if there are disulfide bonds that you need to break, and then your Sanger's reagent actually told you that initially that there were two strands. So, that completes our separation techniques and characterization techniques of proteins. Okay. Okay, now we will go to our next topic, that is this function of proteins, the functional aspect of proteins. Okay. We have completed the structural aspect. Now, we will go to the functional aspect of proteins. Okay. Now, proteins have different functions in our body. One of the major function is, we know that they act as the catalyst, okay, and they catalyze various reactions, and that is, so they have these enzymes. Enzymes is a particular, it's a very important function, but everyone is important here, whatever is written here. See, some proteins are only receptors. That means, receptor means that you have a protein like this, and some molecule binds here, and then that sends a signal after binding. So, but they do not really catalyze any reaction, it is only binding, and that binding is associated with generating a signal. Like what is shown here is rhodopsin. Rhodopsin is the protein that is present in our eye, and then when light falls on it, there is a change in the structure, and actually light is the receptor. Light falls, that is the energy, it disrupts the structure of the rhodopsin, and that creates a signal, and finally we see it, the signal is processed in the brain and we see the image. So, there are then the muscles are also these made up of proteins. Then even the silk that makes the beautiful sarees, that are also made from proteins. Then you have storage materials, ferritin, you have enzymes, as I said already. Then you have transport proteins, which transport, like hemoglobin transports oxygen to the cells. Then you have the proteins which gives us immunity against the invading organisms that affect our body. So, that is called the antibodies or immunoglobulins also called. Then they can be also hormones like insulin, which metabolize the glucose. So, that is the hormone. So, they have different, they have structural, that muscles, they are made up of proteins like collagen, they have this receptor, they have again this is a structural protein, and this is a storage material, this is the enzymes and so on. So, let us out of this we will right now we will discuss the enzymes, proteins which are acting as catalysts. We will do that, and later on we will come back during our medicinal chemistry section to the receptor chemistry. So, these are that is also very important. The similarity between the the receptor and the enzyme is that there are sites which are called active sites, pockets where small molecules bind. Okay. But in receptor there is no reaction, there is a signal that is generated, and in proteins in enzymes there is some reaction that takes place. Okay. So, that is the difference. Now, let us go to the next slide. Now, enzymes, now I know enzymes are macromolecular biological catalysts. They accelerate the reaction. Catalyst means accelerating the reaction. Okay. The enzymes are made up of proteins, that already I have told you, and like all catalysts, enzymes increase the reaction rate by lowering the activation energy. That means, we know that in any reaction, if you want to speed up the reaction, a catalyst, remember, only speeds up the reaction both ways, the forward and the backward reaction, ultimately allowing the reaction to come to the equilibrium at a much faster rate than the uncatalyzed reaction. Okay. But a catalyst cannot give higher yield. Okay. That is a misconcept that we have. Catalyst only lowers the activation barrier, which is directly related to the rate processes of the forward and the backward reaction. Okay. So, basically what happens, I think this is the energy diagram. So, when you have this is suppose the reactant. In case of biochemistry, generally we do not write R, instead of R generally that is called substrate. The reactant is called the substrate, and this is the product P. So, if it is uncatalyzed, suppose this is your activation, this is the pathway, energy pathway that the reaction takes. That means, now the activation energy is for the forward reaction is this. When you have the catalyzed reaction, then what happens? It takes another path where the activation energy is much lower. So, the catalyst must be lowering the enzymes. When they act as catalyst, they are lowering the activation barrier for the reaction, and so that the reaction becomes faster. So, then there is some interesting, still some interesting details here, that the mechanism of enzyme catalysis is like that. The enzyme, as I told you, is like this type of shape suppose, and this is what is called the active site, where the molecule, the substrate molecule comes and binds, and this is what is called the ES complex that is formed, and then the ES complex breaks into product, and then the enzyme is regenerated. To form the ES complex, you have E plus the substrate. So, the substrate goes and binds, forms the ES complex, and then the ES, for simplicity we are not considering the reversibility in this step, that could be also reversible, but the initial analysis of enzyme catalyzed reaction was done by Michaelis and Mentin, and they, for simplicity, what they did that, okay, first it is enzyme plus substrate forms the enzyme substrate complex, that is a reversible reaction K1, and it could be K minus 1, that is the reversal of this, and suppose this is your K2. So, this K2 will ultimately give the product, the rate process for the formation of the or breakdown of the ES complex into the product. Now, because it is catalysis, we know that the activation barrier is lowered, that is for true. But interestingly, when substrate binds to the enzyme, the energy of the substrate also is decreased because the substrate is stabilized. It's like when a baby is alone, the baby is crying, more energy, but as soon as mother comes and take the baby into her lap, the baby stops crying. So, baby is stabilized now. So, it is like that, the enzyme is like the mother, substrate is like the baby. The substrate, earlier the substrate energy was this, and now as soon as it complexes with the enzyme, it forms this ES complex, and that energy has to be lower than this, because I said it is like a that you are stabilizing the whole process, because there are stabilizing interactions. Why the substrate will go here? Because there are weak interactions that stabilizes this complex, okay, like hydrogen bonding or salt bridge formation or or your hydrophobic interactions. So, all these weak interactions play a part in this binding. So, but that also has an activation barrier, because every process needs an activation barrier. So, there is an activation barrier like this, but that is a very small activation barrier, and you form the ES complex. Okay. Now, this ES complex, that means your original energy level now is being lowered. Okay. Then that goes into the ES, that is the transition state. Okay. When the reaction starts, when the bond starts breaking and new bonds are being made. So, at that point, so this is your basically the activation complex, the activated complex the transition state. Okay. Just the transition state is this, and then that goes into the, initially it will be going to the product. At that point, the product is again still bound to the active site of the enzyme, and ultimately the product is released. So, this is also lower than the actual energy of this product, of the product that is released. So, now what is, if I ask that actually between this substrate, the product and the transition state, which one is most stabilized by the enzyme? Because the reaction is like this is suppose the substrate. So, the substrate binds and then it goes into the transition state. So, there is this the transition state where something is happening on the substrate. Okay. So, you cannot write this as substrate, this is the TS now. I think it is, maybe it is not very clear. So, now this will be TS, and that is the transition state is stabilized by the enzyme, and then the transition state will go into E. First it will be E and P, there will be some bonding, definitely some stability, and then P goes out from the active site of the enzyme, and that is then keeps the active site free, and the next molecule comes and binds. Now, out of these three, S, then TS and the product, which is most stabilized by the enzyme? The answer is that it is the transition state that is most stabilized by the enzyme, because this goes down a little bit and this also goes down a little bit. They are slightly they are also stabilized, but to have the lowering of the activation barrier, you need much more lowering of the activation of the transition state than in the uncatalyzed reaction. So, that is the ultimately what I am trying to say is that the enzyme active site must be complementary, most complementary to the transition state, followed by the substrate, and the last is the product, because product is released very easily from the enzyme. Okay. So, earlier usually people used to think that the enzyme stabilizes the substrate, substrate goes and binds. Yes, it stabilizes, but then when it goes to the transition state, that is the highest extent of stabilization of this. We will go to this aspect later on when we deal with synthetic biology. That is why I spend so much time discussing this one. Okay. So, enzymes, okay, these are, I think this we can skip this slide. The enzymes, some structures are given here. Enzymes are proteins. This is a complete 3D structure, complex 3D structure of an enzyme. But let us go to the different classes of enzymes, and how many, like different organic reactions are classified according to the type, like we have nucleophilic addition, we have electrophilic addition, then substitution reaction, elimination reaction. Similarly, based on the type of reaction, the enzymes are classified, okay, and classified into 6 groups. So, whatever enzymes we have in the living world, so they will do one of these reactions. The first class is what is known as oxidoreductase. The second one, what is oxidoreductase? They carry out redox processes, okay, like lactate dehydrogenase. Lactate dehydrogenase is lactic acid going to pyruvic acid. What is lactic acid? Lactic acid is this, and that is going to pyruvic acid, lactate dehydrogenase. Okay. Then the second one is transferase. That means they move a chemical group from one place to another, like an amine transferase. Say the amine group of one amino acid is transferred to another substrate which lacks the amine and puts the amine there. Okay. So, they are called transferases. Then hydrolase. Hydrolase you already have some idea, like the protease, they hydrolyze the peptide bond. So, it could be also other types of hydrolase. So, there is a specific group, that means when your bonds are broken by water, then those enzymes are called hydrolases. So, a specific group is created for that. Okay. And there are many enzymes, lysozyme is one of them, we have chymotrypsin, trypsin, pepsin, so many. Lysozyme, why it is written here, it is quite interesting. The lysozyme is present in our tears, okay. And in our tears, lysozyme is an enzyme which breaks the peptide bonds of proteins, okay. And this lysozyme gives us a very good antibacterial activity in the tear, in the water that is constantly washing our eyes, okay. And then you have, but anyway, hydrolases are ones which breaks a bond by water. Then you have lyase. Lyases are non-hydrolytic bond cleavage. So, if you have a bond cleavage which is done other than water, then you have lyase, those type of enzymes. Then you have isomerase. Isomerase is, there is some isomerization, like say L to D conversion, an amino acid going from L to D, that is an isomerization reaction. And then finally, there is a ligase. Ligase is basically joining of two things together, like synthesis, that means you are synthesis of new covalent bonds between substrates. Okay. And using ATP hydrolysis, because for any synthesis you need some energy, and that energy is provided by this adenine triphosphate. Okay. Now, one important point to note is that if I say what is the different types of enzymes, if you change the order of these classifications, then there is a problem, because the International Union of Biochemists say that if you have an enzyme which is oxidoreductase, that should be given like our IUPAC nomenclature in organic chemistry. So, there is a nomenclature system for enzymes. So, any enzyme which is oxidoreductase, that will get a number 1, initial number 1, and then the species from where it is isolated, and also how many are already isolated from that species, and then the number is put like that. Say if I say 3.1.1, the number 3 means that it belongs to hydrolase. Okay. So, that means you cannot change the order that what is written here. That is very important. So, if something is said at 6.1.2, so that means it is a ligase. Okay. Now, come to this again back to the active site, and what is the mechanism of this, what is the mathematical expression of an enzyme catalyzed reaction. Okay. So, enzymes, as I said, this is the active site, this is one type of active site, and this is your substrate. The substrate has a geometry which is complementary to the active site of the enzyme. But again I warn you that this gives as if the substrate is the best fit, but that is not it. It is the transition state which has the best fit with the active site of the enzyme. Now, another thing that can happen is that so this is also called the lock and key. The enzyme mechanism was earlier described like this, that as if you have this active site is kind of a lock and the substrate is like a key. So, the key goes there, the substrate goes here, and as you do something here, you rotate it, it opens up. So, this is the lock and key complex, and this is the enzyme and substrate complex. And then if you want to open, you have to do something, some energy here, and if you want to get a product, you have to do something on this substrate. So, that is the homology between this lock and key and the enzyme substrate complex. Okay. Now, sometimes or actually frequently it is found that this active site may not be perfectly complementary to the substrate initially. So, what happens, like if somebody we have different body weights and different dimensions. So, if we have a chair, so initially the chair may not fit properly according to your size, but what we do? We ultimately try to feel comfortable by pushing ourselves inside the chair, so that ultimately there is best fit between me and the chair. The same thing happens here. The active site is not really very complementary, there is exact complementarity is here, but as it goes, it ultimately adapts to fit the substrate in the perfect complementary, maintaining the perfect complementarity. Okay. So, this is what is called induced fit model of enzyme and substrate. So, it is not that the enzyme is already having a perfectly tuned molecule. It is like going to a shopping mall and buying a shirt, a shirt. The shirt may not fit that well. So, what we do? We go to the tailor and he cuts little bit here and there, and then it fits to the body very well. So, the same thing happens here. Here the enzyme itself is a kind of a tailor. So, itself it adjusts and then try to fit the substrate into it. So, that is the induced fit model. Now, let us go to the Michaelis-Menten. Before we go into that, the enzymes, there are some of these enzymes, it is not mandatory, but some of the enzymes of those 6 classes, they requires a small molecule for their activity to show, and those small molecules are called cofactor. So, cofactor is a non-protein chemical compound that is bound either tightly or loosely, both are possible, to an enzyme and is required for catalysis. See, that means it is like that you have a big house, everything you have inside, there is refrigerator, TV, everything. You have gone outside and then you forgot or you lost your key. So, if you lose your key, you cannot enter into the house. So, these are cofactors are like the entry point. See, if the cofactor is missing, the protein is made by the system, living system, but if the cofactor is not there, the protein is basically useless. So, sometimes very simple things, sometimes a screw can be a very rate determining step for some machine to be operational. Okay. Types of cofactors. So, cofactors are small molecules which some proteins are they need to show their catalytic activity. There are the types of cofactors are called coenzymes. The coenzymes are basically the organic, if the cofactor is organic in nature, then that is called coenzyme. And there is another one is called prosthetic group. Prosthetic group is basically when it is tightly, cofactor is tightly bound to the protein. Coenzyme is the non-protein component loosely bound to the apoenzyme. Apoenzyme means the enzyme devoid of the cofactor. See, we have said that there is an enzyme like this, there is an active site here, but to do the catalysis you need this small molecule, the cofactor. Okay. Now, the cofactor there are many cases where cofactor is actually outside the enzyme, but at the time of reaction that comes and binds to the enzyme, and then the substrate comes and the reaction takes place. In some cases, the cofactor may already be bound to the enzyme through a covalent bond. Okay. The prosthetic group, the non-protein component tightly bound to the apoenzyme by covalent bonds, is called a prosthetic group. So, when it will be called, the cofactor will be called a prosthetic group when it is attached to the enzyme. Tightly bound means attached to the enzyme. Okay. Now, if you can strip this cofactor off, then this enzyme without the cofactor is called apoenzyme, and when this whole thing is there, that means the cofactor is tightly attached to the enzyme, that is what is called the holoenzyme. Holoenzyme. Okay. And the other thing is that the coenzyme is the organic. If the cofactor is organic, then that is also called coenzyme, but cofactor includes also metal ions, okay, because sometimes metal ions can also be very critical for some enzymes to show their activity. Now, let us go to this, what is the famous equation called the Michaelis-Menten equation. So, the reaction that is there, that E plus S, it is reversible ES, and then the ES going to the E plus product. Okay. And again leaving the enzyme free, and that can again the substrate go and binds. Okay. So, this is the kind of equation that we use, but this should be this should be arrow like this. Okay. And then if you solve this, okay, the first step is E plus S forming the ES complex. So, you have two rate constants K 1 and K minus 1. K minus 1 is the dissociation of the ES, and K 1 is the association of E and S, and K 2 is the breakage of this ES. Usually we put it in a third bracket because this is a transitory, this is not ultimately that is remaining in the medium. It is produced and then broken down either to product or again or revert back to the substrate and the enzyme. So, if you solve this, that is already there, if you solve this equation by assuming the steady state complex. What is the steady state complex? That initially that substrate will go and bind to the enzyme, and then it will also dissociate. This is an equilibrium process, and then a reversible process, and then after some time a state is reached where the rate of dissociation of ES into E plus S and the rate of association of E and S to form the ES, they will be same. So, if that be the case, that means the concentration of ES with time is equal to 0, because if something is constant, that means the differentiation is 0. So, you apply this steady state, and finally, you come to an equation which is this one. Initially, you get this V 0, 0 means actually in enzyme kinetics what happens, as soon as you add the substrate, the reaction starts, and the initial rate, it should go by the 0 curve, the initial rate of the reaction, initially it is linear and then slowly it slows down as the substrate is depleted. Okay. So, V 0 is the initial rate, and that is given by the initial linear line or initial straight line. The gradient of that gives you the V 0, the velocity of the reaction. Okay. So, this V 0 is the initial velocity, that means at the, you cannot have a velocity at time 0, it is very difficult to measure that. So, what you do, you determine some points and say t 1 minute, then 2 minutes, then 3 minutes, and as long as it is linear, you can determine the gradient and get the V 0 out. So, if the V 0 is there, then you solve this equation. I am not going into this solution, you can do it yourself, or any standard textbook have this. V 0 ultimately becomes K 2, K 2 remember it is the breakage of the ES into E plus product into S 0, S 0 is the substrate concentration, initial substrate concentration, E 0 is the initial enzyme concentration, how much enzyme you have taken, divided by S 0 plus Km. Okay. And if you divide it by K, if you divide it by this Km. Now, this Km, before that the equation is like this. Initially you get V 0 equal to K 1 K 2 S into E 0, K 1 into S plus K minus 1 and K 2. These two are added together, and finally, if you divide by K 1, then you get this K 2 S 0 E 0 S 0 plus K minus 1 plus K 2 divided by K 1, and this is what is called the Michaelis-Menten constant. Km is the Michaelis constant. Okay. And so, Km is basically K minus 1, that means the breakage of the ES complex, plus K 2, that means the formation of the product, the rate of formation of the product divided by K 1, that is the rate of association of E and S. Okay. This is the Michaelis-Menten constant, and this equation is what is called Michaelis-Menten equation. Now, you can actually simplify this equation a little bit, that if S is the concentration of substrate is much higher than Km. Remember, this is also an interesting issue, that Km is basically ratio of rate constants, but some of these rate constants are unimolecular rate constants, some are bimolecular rate constants. So, overall it gets a unit which is a unit of concentration, because you have to ultimately add a concentration to this part Km. So, Km has to be in concentration unit, otherwise how can you add one concentration which is S and the Km. So, you can say in both ways that because the equation takes this form S plus Km in the denominator. So, Km must be having the unit of concentration, but you can also do it by plugging in the rate constants, the liter per mole inverse, those type of units you can put, and you will get a concentration unit. Okay. And so, the rate is, if S is much larger than Km, then what happens? The rate becomes, so the equation that earlier you have is V 0 equal to K K 2 into S into E. I am just dropping the zeros, and then you have S plus Km. You can put 0, actually initial enzyme concentration is important. What is varying is the substrate concentration. Now, when S is much larger than Km, then what happens? You can now neglect the Km part of it. If you do that, you will see that V 0 is becoming equal to K 2 into E 0, and that is what is called the V max, the maximum velocity that you can get if you keep the enzyme concentration constant. So, this is V max. Now, you can simplify the equation. Earlier it was K 2 S E 0 by S plus Km. Now, you just write that it will be K 2 into E 0 is V max. So, you can write V max into S divided by S plus Km, that is equal to V 0. So, this is the final form of the equation. Some interesting observations is that when your V 0 equal to half of V max, then if you calculate it, that if you put this value as half of V max, then you will see that Km is equal to S. Okay. I think that will in the next session we will do the further analysis of these parameters. So, there are two important parameters that we learned. One is Km, Michaelis-Menten constant, which is which is characteristic of the enzyme that you have taken, and there is another parameter which is V max, because this will ultimately give you that what is the efficiency of the enzyme. If V max is very high, that means the enzyme appears to be very efficient, or the Km is interesting. Maybe in the next session we will start discussing on the Km. Thank you.